Thank you for the invitation. So yeah, I'm going to spend about the next 20 minutes or so talking about what we've learned from our creatine transporter deficient mice. Can you guys hear me okay? Everything? Okay, so the challenge that we faced was CTD was discovered at Cincinnati Children's by my former division director, Ton DeGraw, and our spectroscopist, Kim Cecil. At that point, we really didn't know much about creatine deficiencies or creatine in the brain at all. There were a couple of papers with the creatine kinase deficient mice. There was a couple of papers with the GAMT knockout mice, but really beyond that, most of the science on creatine was as a sports supplement, muscle function in creatine and all of that. So as a postdoc, I was tasked in developing this creatine transporter knockout mouse. Turned out, as most things with the creatine transporter, it was much more difficult than we thought it was going to be. It took about 5 years to do this, and this went across multiple labs to develop this mouse. And that's because it shares a lot of similarity with other transporters, so it's very difficult to isolate parts of that gene to really target the transporter. But we eventually got it done. So what we needed when we developed this mouse model, when we set out to do it, we wanted a high fidelity model that had cognitive deficits similar to what is seen in humans with CTD. This allows us to test and develop treatments. What we wanted along with this is a model that allows us to answer basic questions about the biology of creatine in the brain. One of the interesting things was when these CDSs, the creatine deficiency syndromes, were discovered, most people thought it was going to be a systemic disorder, something similar to like muscular dystrophy, where you would have smaller sized patients with small stature that had muscle atrophy, as opposed to this profound neurologic deficit, which was very surprising to find. What we were worried about was that not every mouse, every time you knock out a gene in a mouse, it's not going to recapitulate the human. One of the classic is Fragile X syndrome. The Fragile X mouse doesn't have a whole lot of neurologic deficits. It's got a whole lot of behavioral deficits. It's got some key neuroanatomy deficits, but it doesn't necessarily have that strong behavioral phenotype. And that's important because behavior is the primary output of the brain. I'm a behavioral neuroscientist. Behavior is the primary output of the brain as far as I'm concerned. So we knocked the gene out in these mice. We were successfully able to do this, published this in 2011. The first thing that we notice, if you look on this side here, that the creatine peak is gone in the spectroscopy. So that's circled there. You can see the wild-type mice on the top, the knockout mice on the bottom. So there is no creatine peak. There's a small creatine peak, but essentially no creatine peak when we look at this with proton spectroscopy. We did 3 different types of learning, and I'm not going to belabor the types of learning that we do, but like with humans, there are different types of ways that we learn. So we looked at spatial learning, which is a declarative type of learning and memory. These animals have deficits in that. We look at incidental learning, which is novel object discrimination. These animals have deficits in these tasks. We look at state-dependent learning, which is the bottom panel there, and the animals have deficits in this. So this shows that these animals have a nice global cognitive delay, which leads us to believe we've got a pretty good model for this. Oh, perfect. Thank you. Okay. I have to use both hands. So we have a good model. So we then ask, how does the lack of creatine cause these deficits? So for an audience like this, we don't go into the creatine is phosphocreatine, which then is exchanged with ATP. You guys know that, right? So what we actually showed in one of the subsequent papers that we published is that these mice have ATP deficits in every tissue we looked at. So this is an important thing because this gives us, again, an additional target to start looking at mechanisms that may underlie these deficits. Accompanying these decreases in ATP is a compensation by the mitochondria, which is basically the summary of these 4 panels, to try to ramp up energy production in the absence of creatine, which is, has implications for a lot of different things. We also noticed some metabolomic changes. This is newer data. Again, when we did the spectroscopy, we did both protein spectroscopy and we did phosphorus. And what we noticed with the proton spectroscopy, we didn't notice too much with the phosphorus. This takes a lot longer to scan, so this is when we go into the MRS. This was done in a 7T, and as Nicholas said, you have to take your belt off before you go into that room because you'll feel it. But when we look at the proton, we see an increase in glutamate, we see the decrease in creatine that I showed before, and we see this increase in N-acetylspartate. We took this and then we decided to look at this in the whole brain of the animals. So we took the brains out of the animals and ran spectroscopy on it. And we can see the similar increase. So anything to the right is increased in our mice. Anything to the left is decreased in our mice. But we see this N-acetylspartate increase in both. The implication there is that's just sort of a marker of general neuronal health. So then we looked at changes in neuronal morphology. This is through a genetic method where we can label sparse populations of neurons with a fluorescent protein. And basically what we do is draw concentric circles around the soma, around the cell body here. And we just measure the amount of times that that neuron crosses those concentric circles. And you can see that over here where you have less complex arborization in the knockout mice, again, showing that there's probably a deficit in neuronal growth and complexity. We also looked at to further characterize this model, and again, looking for things that we can change. We looked at synaptic plasticity. So this is a cellular correlate to learning and memory, so it's not exactly a one-to-one. But if you have deficits in this thing called long-term potentiation, it's more than likely correlated with learning and memory deficits. So we measured this in the hippocampus of our mice, and it's induced basically by you lay the slice on a grid, you deliver a pulse of electrical stimuli to it, and when you do that, you get this nice increase here in responses to subsequent stimuli. And our knockout mice, again, show this deficit. So what we've got in total here in our ubiquitous knockout mice is a really nice model in which we can start to look at treatment options. We can look at things that we have good readouts for understanding the biology and understanding treatments. So one of the things that we face when we come to treatment difficulties that's unique to the creatine transporter deficiency is this two-hurdle problem. And Olivier Bressant does a really good job of describing this as well. But basically, you have creatine transporter expression at the blood-brain barrier, and you have it in the cells of the brain. So to treat CTD, you actually have to circumvent both of these. So this also raises important questions about what cells in the brain need the transporter. So here are a couple of treatment attempts in the creatine transporter knockout mice that you were all familiar with, I'm sure. The first treatment attempt was in our creatine transporter knockout mice was using cyclocreatine. This was by Joe Clark, using in our mice. And they gave cyclocreatine for 9 weeks, and they see improvements in spatial learning. But this is also a slightly different model than the model I showed before, in that they only knocked the creatine transporter out in the brain. So when I go back, if I go back to this, and I'll show this a little bit more later, this is intact. This is not intact in those mice. So that was one of the limitations of that study. There were other limitations that led to the demise of cyclocreatine that we won't talk about right now. But in the more recent attempts through Aloise's lab, we've shown that DCE can improve incidental learning in our mice. So those are two treatment effects, and this is one of the main goals that we had, was to provide this mouse for treatment. Because obviously we want to understand the biology, but we also want to provide a good model to provide a lot of confidence that when we do develop treatments that they're going to work. So we developed this mouse as a conditional knockout. And I'm not going to belabor this point, but what it does basically is allows us to delete the creatine transporter when and where we want. This allows us to answer basic fundamental questions about the biology of creatine transporter. One of the first questions that we, and I think this morning really highlighted, is we wanted to know, by training I'm a developmental neuroscientist, so I'm always interested in how the brain develops. We wanted to know if creatine transporter deficiency was caused by developmental changes in the brain, or if it was just the lack of creatine in the adult brain that led to these deficits. So what we did, so this is from a graduate student in my lab, we knocked the creatine transporter out in neonatal mice, which is analogous to about the 3rd trimester in human brain development, and we knocked it out in adult mice. And honestly, this was so clean that I double-checked it multiple times. This is amazing how clean this came out. So this is spatial learning. Here we saw similar things with incidental learning, that when you knock out the creatine transporter during development, you get these nice deficits that recapitulate what we see in the whole body creatine transporter knockout mice. But when we knock it out in adult mice, there are no deficits. So this has some implications as we move forward with treatment as well. So when we start to think about gene therapies and stuff like that, the blood-brain barrier problem may not be as significant as maybe we thought it was going to be. That's sort of presumptive, but at the same time could be an important thing as we move forward, is that it does take that early insult. So now how does this happen? We started to look at this where we give this compound called EDU. Basically, it marks any neuron or any cell that's dividing in the brain. And what we see is a general shift in neurogenesis in our knockout mice, where if you look at postnatal day 5, you can see that the knockout mice have less neurogenesis compared to wild-type mice. But on P10, you have this elevation. And then on P15, it goes back down. So it sort of looks like what you're getting is a general shift in neurogenesis that could be underlying some of the changes that we see. And just over here on the right are a bunch of markers that show that developmental genes are disrupted in our knockout mice, which have been confirmed by data from Aloise's lab in organoids. So again, the other question that we have is where? Where are these problems coming from? And the brain is a complex organ, right? So you have a bunch of different cells, you have a bunch of different neurotransmitters, and you have a bunch of different circuits that all basically feed into how we behave. And we focused so far on 3, and then this is the pathways that feed our prefrontal cortex. They're also similar for different areas in which we learn. But we focused on serotonin, which is in blue up here, so from the dorsal raphe to the prefrontal cortex. GABA, which are these red neurons here that inhibit the activity of projection neurons, and then dopamine, which are these green. So serotonin and dopamine are what are known as neuromodulators. What they do is they tune the neurons that they're giving into you're more likely to fire if we give you a signal, or you're less likely to fire, depending on how they process these signals. So those are the 3 that we focused on. You'll see this cartoon multiple times throughout here. A graduate student in my lab, Zuhair Abdullah, he worked on the serotonin-specific model. What we were looking for here, in a way, was we also wanted to know, because the creatine transporter extends just past CTD, creatine's involved in a lot of stuff the brain does. So there's affective disorders, there's Parkinson's disease, there's Alzheimer's disease. We've been asking some of these questions as we go along, just trying to understand the basic biology. He was really interested in how affected, or how it interacted with affective disorders, so he focused on the serotonergic model. Basically, what happened was in the 3 things that we normally look at, there were no deficits. So if you look at the red and the blue, that's the serotonin-specific knockout versus wild type. There were no differences in the serotonin-specific knockouts. We also included our ubiquitous knockouts just to show that we were doing it the right way. But what we did see was in a modeling of an affective disorder, this thing called learned helplessness, where we basically condition an animal to freeze in this chamber, test them a day later by giving them a foot shock and seeing if they escape. An animal that's been conditioned to freeze will not escape this foot shock. That the knockout mice don't remember the foot shock. They go ahead and escape as much as they can. Whereas that should be actually latency, not number of escapes. The wild-type mice freeze normally. The knockout mice escape to this chamber, suggesting they don't remember this. And the conditional knockout mice, the serotonin-specific, also do a similar thing, suggesting that the serotonergic creatine transporter is underlying these deficits. We also have some biochemical data where we looked at these 2 over here are sort of interesting. This is serotonin synthesis and serotonin degradation. This is monoamine oxidase, so if you hear pharmaceutical companies that say when they advertise, don't take this if you take an MAOI, that's MAO. That's what they're talking about. Which could be interesting is this is something that floats around in your blood, so this could be a potential biomarker as well that we could look at. But basically we show that these mice have a deficit in serotonin, both serotonin synthesis and turnover. Again, looking at potential mechanisms of why we see these profound neurological problems. So then we moved on to this GABA-specific knockout. And the GABA, again, are these little red guys that inhibit the firing of neurons. And it's important in this concept of excitatory-inhibitory balance, which is thought to be one of the underpinnings of autism. So when you look into the autism literature, this excitatory-inhibitory balance is a major hypothesis of how autism manifests. So we started to look at these. What we noticed is, again, we always include our ubiquitous, our whole body knockouts as a positive control, just to make sure, again, that the test is running right. That the loss of the creatine transporter in just these red neurons, so the green neuron still has creatine transporter, the red ones don't, is enough to recapitulate these state-dependent deficits. So this is things like remembering place, remembering just certain events that are important in our day-to-day lives. So again, you can see here that they freeze less when they're exposed to a context in which they were presented a negative stimulus. They also freeze less when we give them a tone that is associated with this negative stimulus. So this is important for us to look at how this manifests, because this is a behavior that is somewhat relevant to the human condition as well. Okay, so then the dopamine stuff, there's not a whole lot here, but this is, again, work from Zuhair showing that the hyperactivity is mostly mediated, so these mice are hyperactive, and it's mediated through this dopaminergic system. So one of the things that when we started this is we didn't really know the full phenotype of CTD patients. So we were kind of just guessing as we went through. And whenever we found something, we weren't like, oh yeah, this is definitely something that's relevant to the human condition until actually after we found it for the most part. But ADHD is a significant has a significant coincidence with CTD, so we're excited that we have this kind of persistent hyperactivity, and it is mediated through dopamine. So everything I've shown you so far is pretty solid as far as testing mice. The problem is we don't use any of those tests in human beings. So to measure spatial learning, we swim the mice. To measure incidental learning, we show them two familiar objects, and then we show them something new. We don't really do that in humans, right? So then we started using this test called the 5-choice cell reaction time test. It's a test of attention. The mouse is given a stimulus that they have to respond to. If they get it right, they get a reward of strawberry milk. We shorten the amount of time these lights come on, making the test more difficult and really testing the attentional deficit. So if the animal ignores the stimulus, that's an inattentive. If it selects wrong, that's an inattentive type phenotype as well. So what we show is that our creatine transporter knockout mice have an inattentive phenotype. So really the meat of this is over here. This just shows the amount of time that they're presented the stimulus. But you can see they have an increase in omissions and a decrease in accuracy in this test. They also have an increase in correct latency, so the amount of time it takes to make the correct decision. And again, this corresponds with an ADHD-like phenotype, an attention deficit phenotype. And this is something we want to continue to explore by looking at giving them no-go signals as well and seeing if they just plow through those no-go signals. Interestingly, so first-in-line treatment for ADHD is amphetamine, which is a dopaminergic drug. When we knock out the creatine transporter only in dopaminergic neurons, we do not recapitulate this phenotype. But when we give them amphetamine, which is Adderall, we see an increase, we see actually an improvement. So if you look at the purple bars, anything above the line is an improvement. On this side, if you look at the purple bars, anything below the line is an improvement. So we see a vast improvement in these mice when we give them amphetamine. In fact, if I didn't show you z-scores, you could see that they actually approach wild-type levels as far as the amount of attention they're paying to the task. So this suggests that there's something in this circuit that's disrupted. So when you get these neurons that come from this green, they can terminate on these red as well, especially in things like the nucleus accumbens, and they modulate how well these red cells fire. So we went back to this GABA-specific knockout, and this is relatively new data. I just pulled it together 2 weeks ago. We almost completely recapitulate the inattentive phenotype just by knocking the creatine transporter out in these red neurons. So this is a pretty exciting finding. We're adding more animals as we go along just to show that this is what we think it is. But again, shows that sort of the uniqueness and why we need to do this type of stuff is that we need to think about how we target any potential treatments. 30 seconds. Let's see what we've got. So what we've learned early life creatine is important for cognitive function. This is, again, as we've learned, as we've heard today, I think that this is a pretty concrete idea now. Serotonergic neurons don't really affect memory, but they affect this despair-like learning. The GABAergic neurons, the little red ones, underlie the state-dependent learning and attentional deficits that traditionally get tested when you're doing cognitive testing in humans. That the dopaminergic neurons that feed these neurons, the loss of creatine transport in there can cause hyperactivity, but they don't change attention. And then, the conclusion there is the neurons that receive dopaminergic signals are disrupted. So again, that's these GABAergic neurons. So where are we going? As we've done this now for 13 years or so, where are we going to go with this? One of the big things that we've been facing, obviously, is that we can't restore creatine to these cells. Seemed like, again, everything with CTD, it seemed like a really simple sort of solution up front. We're just going to give them creatine, and we're going to restore creatine, and we can start testing this stuff. But the inability to replace creatine, not only from a treatment standpoint is a problem, but from us understanding the biology is a real problem, because if you can't show that it's the thing you think it is by restoring it, it's difficult to prove that that's what's causing the problem. So, the development of AAV vectors to restore gene function is going to help us advance these studies. We're looking for non-invasive markers for treatment, and we always focus on the behavior of the mice. No matter what happens on a biochemical level, if it doesn't correlate to an improvement in behavior, it's tough for me to get excited about it. So when we talk about viral gene delivery, so just real quick, again, we'll go to this two-hurdle problem. One of the problems is that these cells are always dividing, so every time they divide and you get an AAV that target it, it's going to reduce its expression by 50%. The other problem that we have is the viruses that target these cells don't target these cells. So we have some data that prove this to a certain extent. So we gave different gene therapies or different AAV viral vectors to express the creatine transporter. Top panel just shows that the therapy or that the restoration of the creatine transporter works. This is in Fibroblast. The purple bar shows the knockout fibroblast creatine levels that increases it. We gave this IV to our knockout mice, supplemented them with creatine for 14 weeks, and only got a moderate improvement in total brain creatine levels, and we didn't see any changes in the animal's behavior. We then switched viral vectors to this that's more of a brain-targeted, that only works in mice, by the way. But we gave this just straight into the brain. We, again, don't see an increase in creatine. So really what that suggests is that both of these are required. So when we went to our brain-specific knockout mice, and we took this, again, this brain-specific type of AAV, gave it a retroorbital injection, and you can see that we were able to restore the creatine levels in those mice. This is about 6 weeks after treatment. They were supplemented with creatine. All the mice were supplemented with creatine as well. We left the blood-brain barrier intact and knocked it out just in the brain. So this allows us to provide decent proof of concept on things that could work. It is way upstream of any potential sort of human trials or anything like that, but what it does is it gives us the chance to say, if we can restore the creatine transporter in the brain at a given time, then we've got confidence that it'll work, or we don't have confidence it'll work. But it gives us the real chance to test this. And as I put this together, I kind of racked my brain on why it took me so long to get to this instead of just plowing through in the ubiquitous knockouts. But it is what it is. Finally, real quick, as far as non-invasive ways of treating or of measuring, this is magnetoencephalograms. So basically, anytime you have an electric signal, you have a magnetic signal. And we can measure this magnetic signal in mice. I don't know anything about this. This was collected via a collaborator of mine, but I put what he said to me is that they're very interesting. They have a more diffuse signal. It may indicate something. I'm not sure what that is, but it's something. The development of the brain associated with clear signals, so these sharper lines. For human disorders, patients with autism may have a more diffuse signal. So again, you see that there. So this could be a good marker for us moving forward as well. We're still collecting these data, but it's pretty exciting so far. And with that, those are all the acknowledgments of people that have helped do this work. Obviously, the biggest thanks goes to the ACD for organizing all of this stuff and really just helping guide what I'm trying to do to tailor our models to fit what we see in our patients. So that's what I've got, and I'm happy to answer any questions.
Okay, thank you, Dr. Skelton. That was a wonderful talk, and all right, I already see questions. I'll open up to questions from the audience first.
Thank you for the presentation. Are you doing males and female?
We have done both. We primarily focus on the males right now, but we have shown that the female mice, I didn't include it in here, but the female mice have a in-between deficit. So they have about 50% of the brain creatine, but they also, let me go back to the microphone instead of yelling. They have about 50%. They show deficits, just really small deficits in spatial learning and incidental learning, but didn't show that sort of state-dependent contextual fear and freezing. Part of that is exon activation, and when we start getting into conditional knockouts, it's very difficult for us to control for that exon activation. So if you've got a gene, you're expressing two different genes, and you're trying to target a gene that may be completely wrapped up in histones, there's going to be so much variability between each one of those female mice. I mean, think of calico cats, right? It's the different spot patterns, basically. It's just been one of those, it's a little bit beyond our bandwidth, is really what it comes down to, is we would really, really need to increase the amount of mice that we test to overcome some of that variability. Not that I'm opposed to it, but we would just need more people and more.
Well, if you were able to, then would you? I think you'd almost have to start from scratch in terms of what behavior is affected, and maybe some of the things that you ruled out in the male mice, you might need to reconsider and all that kind of stuff, right?
Right. Yeah, and it's always been, for better or for worse, we've always had that assumption of if we can get the males online, the females will follow along. And I mean, it's not the cleanest way to say that, but that's always, we're going to go with the most severe phenotype that we can, and hopefully the less severe phenotypes match as we go. So
I know that feeling. We're in that same feeling. I'm not sure it's the right feeling, but it's a reasonable feeling, right?
Yep. The NIH reviewers also don't think it's a reasonable feeling.
That was great, Matt. I didn't see the details of the Cree drivers, but given the effect on neurogenesis that you showed from doing the early versus late knockout, do you think that the lineage-specific knockouts are affecting sort of early neurogenesis at the point of specification, or do you think that there is a role later on?
No, that's a great question, and it's really complex, right? Especially when you think of both, not to get too drilled down into the details, but both serotonin and GABA are very important for brain development. We picked drivers that when the brain is developing, when a cell decides it's going to be a serotonergic neuron, it loses the creatine transporter. Right. When a cell decides it's going to be a GABAergic neuron, it loses the creatine transporter. That could affect the way that the neurons of the prefrontal cortex or the hippocampus develop, because both of those, getting signals from those neurons is really important for the way that those develop. So I guess the thing that gives me a little bit of hope on the specificity is we didn't see a ton of Morris-Watermaze deficits. So that's a hippocampally mediated. So if the development of the hippocampus was being disrupted, it would have manifested as a deficit in that spatial learning. Yeah. So that, again, it gives me some hope that we're actually targeting the neurons and not necessarily sort of a secondary effect.
And the phenotypes are specific.
Yeah, and it's very cool. I mean, I was expecting messy, and it's actually been pretty clean.
Nice work.
Thank you. One question on the mice that were treated with AAV, because it looked like the creatine level was higher than the wild type. Mm-hmm. Did you see any toxicities or any behavior issues if you test them with the behavioral test?
So we haven't tested those guys behaviorally yet. We didn't see any toxicity issues, so we let those guys go for a while, just that sort of thing, right? And we did not. And again, I don't know if that's because we have good regulation at the blood-brain barrier where we're not getting a ton of excess creatine flooding in, so there is that level of regulation. And again, that's why this is a one-off of any sort of preclinical gene therapy testing. This is more of we're going to answer basic biological questions with this model and have it ready to go when the technology evolves to the point where we can actually do real testing where we can address both the blood-brain barrier and the neurons. So
Wonderful data. I have to say, wonderful data. Now, when you injected into the brain, you said, when the brain means into the parenchyma, into ICV or ICM?
ICV.
ICV.
Yeah.
And at what age?
Those guys were P28 to P42. So we weren't going to try to do any sort of proof of concept on behavioral. We just wanted to see, could we get creatine in the brain? We sort of, on the AAV, the first AAV9 study, I kind of learned my lesson. Let's look at creatine levels before we look at behavior, because we did a bunch of behavioral testing on those AAV9 animals, and it turned out that the brain creatine levels weren't there. So we did a lot of work to have mice that look like knockout mice, essentially. So yeah, right now it's just more, what's the best targeting to get creatine into the brain?
All right, I think for time, this will be the last question. So here's this.
Hi, Matt. Thank you. It was very interesting. Could I ask you a couple of questions on very briefly on a couple slides back here? So this mouse is where you've knocked out creatine transporter only in brain cells, not the blood-brain barrier. Is that correct?
Yes.
And the promoter that you use then, is that a neuron-specific promoter, or that's a like
It is a ubiquitous promoter.
It's a ubiquitous promoter.
Ubiquitous
Okay.
promoter. But it was the same promoter that we used, same virus basically that we used in the bottom in panel C.
Thank you.
Yeah.
All right, thank you so much for that.